Good morning, everybody. Welcome to Biodiversity Genomics 2023, and particularly for this Thursday about Biodiversity Genomics Europe Day. It's my privilege to be your chair this morning. My name is Gilles Doignon. I'm team leader Biodiversity and Nature-Based Solutions at DG Research and Innovation at the European Commission. The BGE Consortium is an EU-funded project and consists of 33 partners from 20 countries. And today's event marks the end of the first year of the BGE project. The BGE Consortium brings together two newly formed European networks: BIOSCAN Europe, focusing on DNA barcoding to understand and monitor biodiversity at the species level, and the European Reference Genome Atlas, focusing on the production of reference genomes to understand and monitor biodiversity at the genomic level. The structure for today, this Biodiversity Genomics Europe Day, is a first session, so this one, to give an introduction to the biodiversity policy landscape in Europe, to know more about the BGE project and the details of the barcoding and genome sequencing streams. Then there will be two subsequent sessions of contributed talks featuring case studies, the first focusing on using genomes to understand and conserve biodiversity, and the second focusing on DNA barcoding approaches for biodiversity characterization and monitoring. The penultimate session of the day focus on invited talks covering recent innovations, which offer opportunities for increased utilization of genomic data for biodiversity characterization, conservation, and biomonitoring. And then the day today will finish with a panel discussion exploring what do we need to do to maximize the impact of major biodiversity genomic projects in meeting stakeholders' needs. So I will start with the first presentation with an introduction to the European policy landscape, the priorities for the European Commission, and the landscape in which the BGE project sits. Then my friend Dimitris Koureas, Managing Director for Digital at Naturalis Biodiversity Center, but also the Director of BGE and the Executive Director of DISCO, will connect with what I said and the connection with BGE, outlining the rationale for bringing together barcoding and reference genome sequencing, the links to other infrastructure in Europe, and the overview of the BGE project structure and approach. Then my new friend, Peter Hollingsworth, Director of Science at the Royal Botanic Garden in Edinburgh, will speak about the DNA barcoding stream, how it fits with the International Barcode of Life, BIOSCAN Europe, and then the barcoding stream specific objectives and what has been achieved in the first year of the project BGE. And then another new friend, Camila Mazzoni from the Leibniz Institute for Zoo and Wildlife Research, Berlin, will speak about the reference genome stream, how it fits with the Earth Biogenome Project, the European Reference Genome Atlas, and then the genome stream specific objective, and again, what has been achieved in the first year of BGE. After my presentation and the presentation by Dimitris live, there will be the two recorded presentations by Peter and Camila, and then you can send questions. There will be time for question and answer at the end of this session. But I will start with my own presentation, and I will then share my screen. There you go. And so I wanted to give to take a distance before we go directly to DNA, and my presentation is about the EU and the global biodiversity framework. And yeah, with genomic science, we are opening a new chapter for science, but also for nature. So just to remind you that we are facing two major planetary crises, and many people know about the climate crisis, but the nature crisis could be even worse. IPBES is telling us that 1 million species are at risk of extinction within decades, and this sixth global extinction is actually the result of our own activities. And the main causes of biodiversity loss by descending order of importance are changes in land and sea use, for example, urbanization and agriculture. Then direct exploitation of organisms, such as fisheries or hunting. Climate change, number 3, but increasing. Then pollution, and last but not least, invasive alien species. So what do we need to do on this planet is really to protect, restore, and sustainably use biodiversity. If we do not, we are doomed. And this is really at the core of the EU Green Deal, and particularly its biodiversity strategy, and it concerns all sectors. And of course, it is related to the global biodiversity framework adopted at the CBD COP15 last December. I really wanted to make a reference to one of my professors at university, Raymond Rasmont, who used to say, it is impossible to know something you do not know. And so actually I will rephrase by saying it is impossible to protect, restore, monitor, or sustainably use something you do not know. And this is why for centuries we have taxonomy. So really to ensure what is nature, to describe it. And of course, taxonomy evolved a lot, and now we are speaking of all aspects of taxonomy, from collections to DNA, digitalization, and this is where the EU funded or is funding many projects, such as DISCO Prepare or Synthesis Plus. There are different aspects like access to scientists and policy, and also to the whole society. This was the Mobilize project. The importance of citizen science, where actually we can get a lot of information about population. And we have this very important Tetris project ongoing. Also, to mention that we need infrastructure, so data research infrastructure for biodiversity, such as DISCO or ELTER. And also DG Environment launched a specific project to build a new generation of taxonomists, because many young people think it's boring. Well, actually it's not. And so this is why there is a specific project about the red list of taxonomist project. And what is very important is really to link local taxonomic knowledge and expertise, for example, near biodiversity hotspot and protected areas, but not only. It's really about biodiversity everywhere, also in cities, and we need to have impact on the ground quickly. Probably you have seen the paper by Richardson et al. In Science last month in September. About all the planetary boundaries, and 6 of them are not good. And you see with the arrow that actually it's really about the genetic richness that we are losing. So we are in real trouble in terms of genetic information and richness on this planet. All this is related to the global biodiversity framework, particularly the Global Biodiversity Framework Target 13, which is about fair and equitable sharing of benefits from genetic resources, digital sequence information, and associated traditional knowledge. But actually, genomics are also related to 6 other targets, target 1 to 6. Target 1 and 3 are really about effective conservation of biodiversity, in which species diversity and genetic diversity is implicit. the Global Biodiversity Framework Target 4, covering the genetic diversity angle as well as species diversity, the Target 5, focusing on sustainable use, harvesting, and trade of wild species, which encompass species and genetic diversity. And finally, the Target 6, where DNA biomonitoring and detection of invasive species is very relevant. So you see, genomics are very important. This is why we are here today. And also, this is why we have funded this Biodiversity Genomics Europe project. The Biodiversity Genomics Europe project is not alone, so there are other EU-funded projects focusing on genomics or using genomics. So I put some examples, and I hope that my slides will be shared. I just want to mention that right now at the European Commission, we are thinking about building a new cluster of projects. So the project really focusing on taxonomy and the project focusing on genomics. It's not confirmed yet, but this is what we are thinking right now, really to ensure that we have no duplication and really we ensure we have synergies between all scientists. I just wanted to show this very complex slide. Actually, it took a lot of time to make this organigram of who does what at the EU level in terms of biodiversity. And so following the EU biodiversity strategy and also linked to the global biodiversity framework, we are trying at the EU level to have a clear way of who does what with who, when, for what. On the left, you will see that there is a new big body called the EU Biodiversity Platform. This is where all the European Commission Directorate General's stakeholders organization are meeting to discuss policy needs and knowledge gaps to be filled in. And these are sent to, in the center, the Knowledge Center for Biodiversity. This is a key body created in 2020, so the KCBD, coordinated by the GRC. And this is where all the questions about research on biodiversity are discussed using different tools. And in the middle, you will see that there is also the Science Service for Biodiversity, coordinated by the project BioAgora. This is where, when the GRC is unable to give a quick reply, the Science Service for Biodiversity is activated and we look for the right information where. And this is then when we go on the right with the different sources of information. There are many important projects, and for example, for genomics, BGE, this is where I put an arrow, is under the projects coordinated by the Research Executive Agency, but DG Research is the one deciding the topics and project to be funded. I will not go in more details about that, but this information will be soon available with a lot of explanation on the GRC website. And then my last slide is just to say that we are preparing the future. So at DG Research and Innovation, we are working on the strategic plan 25-27. So really to identify the knowledge gaps and priorities to be funded in the last part of the work program of Horizon Europe. We already started to work on the next framework program, the FP10 for 28 to 2034. But also in our unit and with my team, we are working on the long-term strategic research agenda for biodiversity. So this is really to have a transparent way to explain to everybody which should be the priorities by 2030, 40, 50. And so, for example, what I put to really have a complete overview of the biodiversity status from space to genes. So, for example, there are international initiatives developing right now, like the Geo Global Ecosystem Atlas, and also you have many projects with the LIFE program on the ground. We really want to include everything so that we know what the EU should fund and what the other could fund, like the member states. What is very important is that our work should follow and feed the work of IPBES. So really that the EU research will provide the information IPBES needs and that the EU research will adapt with the needs identified by IPBES. For example, with the latest assessment adopted this year on invasive alien species, the use of genomics. And also, we are analyzing all needs regarding biodiversity, so including infrastructure. So that was it for me. And I will now give the floor to my friend Dimitris. Thank you.
Thank you, Gilles, very much. Let me see if I can share my screen. Yeah, thank you very much. Thank you, first of all, for your sharing the session and the day today. And thank you very much because you have very wonderfully, I would say, set the scene up or described the scene in which the Biodiversity Genomics Europe project is called to operate in. You quoted your professor, Rasmon, saying that we cannot know what we do not know, and I think that's at the essence, basically, of many of the activities of the community of biodiversity scientists. And I would say that despite the advances we've made in biodiversity studies over the years, we're still falling behind in describing biodiversity, but also understanding and modeling it in a way that we can inform more effectively decisions for replenishment, preservation, and sustainable exploitation of our natural resources. And of course, I borrowed this slide from you because I think it's a wonderful illustration on that three waves that you showed also in the beginning. And of course, the integrity of the genetic pool of this planet, it is at a huge risk. Effectively, documenting the planet's vast natural genetic resources before they vanish is not just important, is a crucial mandate that we have from society towards the global scientific community. And I would argue that we shouldn't be fooling ourselves. This is a fight against time. And it is a fight in which we need to mobilize and organize the entire landscape as effectively and efficiently as possible and bring in any available resource to contribute towards that huge battle. And I think this is exactly the premise or the context, if you wish, in which the Biodiversity Genomics Europe project is called to operate, and exactly the premise that has driven our approach up to now in putting this project together. I'm extremely thankful to the European Commission because it offered the space of opportunity for us to be able to do so. And I hope that this project will start bringing into play some of the new capabilities and joint capacity that we can have at the European scale. So as you mentioned, Gilles, the Biodiversity Genomics Europe brings together the DNA barcoding and the full genome communities in Europe for the first time at this scale together. It is a joint endeavor that is uniting these two major strands and fundamental strands for biodiversity genomics research, and sets, to some extent, hopefully, the foundations for a more massively scaled up delivery of DNA-based evidence for biodiversity. And I think that's the key message I would like to convey to the audience, because we will have the opportunity to hear from Pete and Camila in the next two presentations more details about the content of those two strands within the project. The project involves currently 33 partners across Europe. But more importantly, I would say, through the vast networks of the two communities that are coming together, I think we are connected to more than 200 organizations, basically, in Europe. And I think that's exactly shows the, let's say, the role that the project plays and is willing to play within the European landscape. BGE has a dual mission. On one hand, on a scientific perspective, to accelerate the use of genomic science, to enhance understanding of biodiversity, monitor biodiversity chains, and guide interventions to address its decline. So there's a very specific part of the project that aims towards delivery of content of knowledge at scale. But there is another part of the project that is all about how it affects change on a systemic level, and how we bring together national and existing institutional level initiatives all across Europe in order to build those economies of scale and scope that we need in order to deliver, in turn, impact. And we're trying to do this, all of that translates into three key objectives that spans from what we call the capacity pillar, the production pillar, and the application pillar. In the capacity pillar, our fundamental aim is to establish, as I said before, a functioning biodiversity genomics networks at the European level to connect and grow the community capacity and to tackle the biodiversity crisis using genomic tools. We have, of course, a mandate to build and continue enhancing the European nodes for both iBOL and EBP, the two large international initiatives, projects that need to find their way within the European landscape and connect all of these European national nodes together. There are a few other ambitions that you see in the slide, but as I said before, you will hear more about the content in the following presentations. On the second pillar of the project, we're trying to look into the production, basically the 20 countries that are working together within the project by connecting samples to sequencing facilities, to data management, and from data management workflows to delivering impact for science. So we have a very concrete value chains within the project that basically start from the going out in the field to delivering impact on specific use cases in the project. And you will see how this translates into the structure of the project as well. And of course, the application layer is that end of that value chain within the project where we are trying to deliver using DNA barcoding and genome sequencing technology to enable biodiversity characterization and conservation for biomonitoring in taxa and systems of high conservation, economic and socioeconomic importance. So we have made a few, we have selected a few cases where we can demonstrate the value of how we, by interconnecting all of those elements, all of those organizations, all of those people, and with their expertise across Europe, we can actually accelerate the delivery of knowledge like we have never done in the past, at least within the European landscape. All of that translates into a structure of the project. I'm not going to go, of course, into the details of the work packages, but what I would like you to see here is essentially the structure. You will see two big strands that horizontally transcend the project, the iBOL or barcoding stream and the ERGA stream or the whole genome stream. And there you will see also what I said before, the value chains coming from the sampling, the sequencing, the data and the application layer, and how all of these work packages are hugely dependent on each other to deliver that chain of value. What I would like to highlight here is three things. One is that consolidated effort in the middle of the slide, where basically is a big part of the project financially in terms of the resources on how we try to deliver common knowledge, common resources, common services for the two communities. And it's an opportunity to start for the two communities to start coming closer together now and hopefully more in the future. You will see on the left-hand side of the slide, work package 14, which is a policy and ethics, a very, very important and to some extent quite painful after now, I would say, part of the project, how we navigate the complex world of ABS and the Nagoya Protocol, especially when the project is called to operate across countries. And as you all know, this already means that we need to go through this very complex landscape, policy and legal landscape. Coming towards the end, I would like to bring back one of the slides that Gilles presented, this complex landscape that very nicely the Commission is trying to organize. And I would say, of course, that not only BGE, but projects like BGE that start at connecting initiatives and developing these economies of scale and scope are fundamental in that knowledge provision section of that slide. And of course, this can happen through multiple vehicles of delivery, either through European projects or in the future through more sustainable organizations such as research infrastructures. But the bottom line is that the communities themselves have a very important role to play on how they self-organize and how they streamline and funnel all of that effort so that we can offer that knowledge, that necessary knowledge to the science policy interfaces that the Commission, but also at the global scale, were in need. I will close by saying that, of course, the project is not only looking into the European perspective, but we believe that it is also a building block towards a more international or the global aspirations that we all have set for ourselves for the future, to deliver a global library of barcodes to discover and monitor biodiversity, and of course, a global atlas of all species genomes, which are the fundamental principles on the iBOL and the EBP international initiatives. So I hope that the project, as Gilles said, is finishing its first year. And over the next two presentations, you will be able to hear a bit more about the content delivery in the two major streams of the project. So I will finish here. And thank you very much, Gilles, for the opportunity.
Hello, I'm Pete Hollingsworth from the Royal Botanic Garden, Edinburgh. And as part of this introductory session to the Biodiversity Genomics Europe Day at BG23, I'm going to give some details as to the plans, ambitions, and initial progress of the DNA barcoding stream of BGE. So just to give some orientation, the BGE project supports the European endeavours of two major global programmes. So the International Barcode of Life, or iBOL, using short, standardized regions of DNA for high-throughput species identification, and the Earth Biogenome Project, focusing on the large-scale production of reference genomes, providing insights into genetic and genomic diversity. So as I'm focusing on the barcoding side of BGE, I'm going to start by summarizing the aims of the global International Barcode of Life. So the iBOL program is a major international collaboration led by Paul Hebert from the University of Guelph in Canada. And its overriding motivation is, on the one hand, the fundamental importance of species as a key element of biodiversity. And on the other hand, the general difficulty of telling species apart at scale and understanding how species respond to environmental change. And so the iBOL consortium has made remarkable progress in building a DNA-based index of life on Earth, including the accumulation of more than 14 million DNA barcoded specimens representing over 1 million different taxa. And this reference library supports species identification and species discovery from individual specimens, as well as biomonitoring of species from the sequencing of bulk samples through DNA metabarcoding, and of course increasingly from sequencing eDNA, the traces of DNA detectable from species in environmental samples such as water, soil, and air. So supporting this global international work of iBOL, we've initiated a European node of iBOL called BIOSCAN Europe. And the aim here is to provide a mechanism to connect and strengthen the multitude of national activities, individual researchers and projects in Europe that are working on DNA barcoding and metabarcoding. And BIOSCAN Europe started with a minimal resource base and somewhat organic origins, but has since received a major injection of energy and an increase in pace of activity via the Biodiversity Genomics Europe project. And so this schematic here summarizes the barcoding stream of Biodiversity Genomics Europe. And this includes resourcing the development of the European node of iBOL, as I've just mentioned, as well as establishing workflows, infrastructures, and data production pipelines, such as sampling in museums and in the field, and taking those samples through barcode sequence generation, data processing and analysis, and then linking the resulting data through to practical applications. So, in a little more detail, that includes establishing a governance framework and growing the reach of the European node of iBOL, but as well as working at a continental European scale, also working at a national scale, so sharing ideas and experiences on how best to set up national barcoding campaigns. There's also work aimed at filling in key gaps in the European barcode reference library for species of particular importance for biomonitoring, such as pollinators and species relevant for water quality monitoring. There's also a particular focus on developing pragmatic approaches to clean up and curate the existing barcode reference libraries for key groups in Europe. And there's a complementary strand of work undertaking biomonitoring across different systems, and a strong focus here on working to harmonize approaches across these different systems. So other key objectives include refining dark taxon barcoding protocols, so using high throughput barcoding to enhance understanding of diversity in megaspecies rich groups, and more generally aiming to enhance protocols for making barcode sequence recovery more reliable and more cost effective. There's also a strand of work evaluating options for multi-locus DNA barcode approaches in groups like plants, where the standard barcoding approaches often do not provide species-level resolution. Then from an informatics perspective, a key task is establishing a European instance of the Barcode of Life data systems, so the big barcode database, and also enhancing the suite of tools for handling DNA barcode data. And finally, there's a very strong citizen science element to the BGE barcoding work, and this includes general public involvement in contributing to biomonitoring, as well as harnessing the expertise of specialist amateur experts for building and curating the barcode reference library. So it's been a hectic startup year for the project. We're just at the end of the first year, and there's been already a lot of progress against these objectives. And just to highlight some of these, that includes extensive consultation and a workshop on best practice for building national barcoding capacity, and the application of this in test cases, building national barcoding programs in Poland and Greece. The metadata standards for the project have been established, and the European instance of BOLD is now set up and operational, and that's been a great achievement. We've also completed a first iteration of a user needs assessment to guide future informatics development work for barcoding, data handling, and analysis in Europe. Also completed a first pass gap analysis for the European barcode reference library to really prioritize sampling efforts. And alongside that, we've developed a conceptual framework for driving forward the barcode reference library curation. Work at the Natural History Museum in London has made substantial progress on honing effective and reliable genome skimming approaches for routine recovery of DNA barcodes from museum specimens to support reference library construction. And in parallel, there's been extensive fieldwork across Europe, including field collections to strengthen the reference library representation of aquatic invertebrates to support DNA-based water quality monitoring. Malaise traps have been set up and run along elevational gradients across Europe's major mountain ranges for insect biomonitoring, and this is to establish baselines for biomonitoring responses to climate change. And likewise, soil sampling has been undertaken at sites across Europe for DNA barcode-based biomonitoring of soil arthropod diversity across gradients of forest restoration and regeneration. So that was just a very brief whistle-stop summary of the DNA barcoding workstream of the Biodiversity Genomics Europe project and some initial activities in the first year. Over the next few years, it'll be very exciting to see further progress of the project, and many thanks for your attention today.
Hi, my name is Camila Mazzoni. I'm a scientist at the Leibniz Institute for Zoo and Wildlife Research in Berlin, Germany. I'm here to talk on behalf of the genome stream of the Biodiversity Genomics Europe project, one part of the project that is tightly linked with the European Reference Genome Atlas. In this presentation, I'm going to start talking about the Earth Biogenome project, explain a bit about the ERGA network, finally describing the goals and some of the highlights reached by the genome stream of the BGE. The EBP is more than a project, it's a network of networks that connects networks and projects all over the world. The EBP includes, for example, two major projects such as the Vertebrate Genome Project and the Darwin Tree of Life, as well as two major regional nodes, the European one, or ERGA, and the African Biogenome Project. Different publications came out in the last years announcing the mission of the EBP and explaining why we should sequence all eukaryotes on Earth. Of course, having these resources will revolutionize our understanding of biology and evolution, but it will also help us make informed decisions for conservation, protection, and restoration of biodiversity, finally creating new benefits for society and human welfare. The process for establishing genomes for each eukaryotic species is a multidisciplinary one that starts with sampling in the field, followed by taxonomic classification, extraction of high-quality DNA, sequencing using specialized technologies that creates data that is then used for assembling, curating, and annotating the genome. And the handling of this data is then made available openly in an accessible way for finally being used for analysis that can touch many different fields of science. This is, of course, a major challenge. Working in a multidisciplinary workflow, trying to sequence an enormous amount of species, this must be made and coordinated at an international level. About 3 years ago, the European scientific community started a movement to self-organize as a massive network of scientists with the goal to coordinate the production of genomes of the European biodiversity. This movement gave rise to the European Reference Genome Atlas, or ERGA Consortium, which counts with a governance structure that includes representatives of the different European countries, as well as committees of experts that cover all the aspects of the genome establishment workflow, including those technical, ethical, and of dissemination. The committees work to find the best strategies to promote the sequencing of high-quality genomes at a pan-European level in an inclusive manner. In order to test this decentralized model at a continental level, ERGA has run a major pilot project, a 2-year-long endeavor that involved most European countries and tested a distributed sequencing and computational infrastructure, establishing then this way the first ERGA resources in terms of policies, SOPs, and code of conduct. Which allowed the collaboration among ERGA members, institutions, and also with biotechnology companies. The pilot has given ERGA the basis to plan and start the European-funded BGE with several of the logistic challenges already recognized. The BGE is a joint effort of ERGA and the European node of the International Barcode of Life, known as BIOSCAN Europe. The BGE has two independent streams, the barcodes and the genomes one, which are connected by pillars that represent the major steps to generate and analyze biodiversity genomics data. In this presentation, I will focus on the ERGA stream of BGE, its goals and outcomes reached in the first year. So the genome stream aims to establish and implement large-scale biodiversity genomic data generation workflows. These workflows are used to generate reference quality genomes from critical European biodiversity, as well as biodiversity hotspots, pollinators, and a selection of applied case studies. One of the main missions of the genome stream is to support ERGA in its mission to accelerate the production and accessibility of reference quality genomes for species across entire European biodiversity. During our first year, we have promoted the participation of the community through different calls, such as the second call for participation of the community to provide samples for reference quality genomes, which was then associated with the development of species prioritization guidelines for genome sequencing. We also developed SOPs for specimen sampling, metadata collection, and shipment that were made available publicly in our webpage. Another call that we launched this year was for selecting hotspots, areas of hotspots of biodiversity for sampling for reference genomes. This call has finished the first phase, is now going for second phase with 6 amazing biodiversity hotspots areas pre-selected. Also to guarantee that the samples, the high quality samples are going to be preserved as well as their filters, we have defined and developed biobanking standards and guidelines for the projects that are linked to the Global Genome Biodiversity Network. One way to follow all of this complexity and decentralized infrastructure for sequencing and computing, as well as sampling from all over Europe, was to develop an internal tracking system made by our colleagues at STENAG. Also, for data to applications, we have been developing workflows for assembly and annotation. We have already released tutorials and training materials for manual curation of genomes, and the ERGA public portal that displays all the public available data that is connected under the ERGA umbrella has been released. Also, as part of our first year, we have launched a call for extra case studies from the community that will increase the number of species connected to conservation, bioeconomy, and disease, showcasing the application of reference genomes for applications to the society. With this, I hope that I have given you good overview about our work in the first year of the project and the goals, and I thank you very much for your attention.
Thank you very much, Camila, Peter, and Dimitris. And well, personally, I have to say it's great to see the first development in the first year of Biodiversity Genomics Europe. We need to do more. We will do it together. As a chair, I'm now checking the question that we have received. There is only one question from Rob Waterhouse, and actually it's for me. So I will read the question by Rob. Given the commitments of countries to report on the COP15 agreed targets and their associated indicators, already are European countries to be able to deliver meaningful metrics? And what is the Commission planning to specifically support countries in this endeavor? Well, actually, this is more a question for my colleagues at DG Environment following the implementation of the Global Biodiversity Framework. For DG Research, we are more in, how to say that, in support. So what I know is that right now the different countries, including the member states, are preparing the national biodiversity strategies and action plans. This is also where we will get information about their needs, but it's also where a project like BGE, but then also many important organizations such as CETAF, the Consortium of European Taxonomic Facilities, will help us to find the best way to help countries or researchers. I see more questions coming for me. Oh, yeah, more and more. So one from Claudio. As the European Commission envisaged or already implemented a path whereby genomics plays a key role as one of the parameters in the biodiversity policy-making process. As far as I know, not yet directly, but the fact that we funded BGE shows that, yes, we are going in this direction. And this is, at least at DG Research, what we want to discuss with other projects and then go back to the EU biodiversity platform. Then I have a question from Mark Blackster. How will the dark taxa be included in ERGE? This is maybe more a question for Camila. I don't know, Camila, if you could reply on this.
Yes, sure. So we have, in this first phase from ERGA and the BGE, we have developed a prioritization species guideline or procedure for that. That includes taxonomic representation as well from the phylogenetic, the eukaryotic phylogeny point of view. So this is going to then give more weight, and over time, as certain taxa get less represented in the genome databases, this is going to give more weight, so that's the so-called maybe dark taxa, or the one, or maybe if they, okay, let me answer about the ones that are underrepresented, because maybe what Mark is talking about when he means dark taxa are the ones that are not yet identified. And so this is, I think, a very important connection that we're going to have with the barcode community, because they are on the front line for the discovering species and biodiversity identification, while we follow up with the genome sequencing of those species that are already known. So we have this both then sites, the one that is for underrepresented species and the ones for the species that are not yet known. I hope this answers the question.
Thank you very much, Camila. I hope it is indeed the reply interesting for Mark. I can also add that we speak about ugly species where people do not want to work on because they are ugly, but actually we need all species. I see that we have still 3 minutes, so I will take 2 questions. Oh, sorry, there are more coming, but there is one question from Jose Alonso. Could you please elaborate just a bit more on the idea of developing a cluster or project of genomics? Yes, and this is related to the question by Christian de Goutry. How does the European Union plan to promote and facilitate internal efforts within genomics-based projects to connect and collaborate independently, fostering synergy and data sharing among these projects? This is exactly the idea of this cluster. So we have different clusters, and for example, in March, we launched a cluster about transformative change with 11 projects focusing on transformative change, or not necessarily, but actually they are working on it without naming it. So it was very, very cool because the different projects, we did a kind of speed dating. So each project saying, this is what we do, this is what we need, this is what we can offer to others. And we saw that the different projects really started to work together. So the idea right now, but not confirmed, we need to check in terms of logistics, is really to launch a cluster specifically about species conservation, monitoring, identification. So the pure taxonomic work, but also the genomic work, citizen science. And so right now we are listing the different projects that could join that cluster, so including BGE and others, but really to have all these synergies and avoid duplication. So I hope I would be able to confirm that within 1 month, but I need a green light from different colleagues. And then I see there are other. Okay, I will have a last question maybe from Donat Agosti. Taxonomic names are based on published taxonomic concepts, allowing to compare sequences of a species over space and time. Wouldn't be important to provide access to the estimated 500 million pages of scientific literature, itself a huge challenge to convert into digital accessible knowledge? Well, then maybe Dimitris or Peter, you would like to take this one? And you are more expert than me.
Yeah, of course. Thanks, Gilles. I absolutely agree with Donat. Of course, the challenge here is to interconnect all the available knowledge that we have and data sources and data types that we have for biodiversity. And of course, original descriptions, treatments are fundamental part from that. And I think we are very happy that we have infrastructures like the treatment bank that actually mobilizes all of that information. The challenge still lies on how we make sure that all of that is fully interconnected and available through any point of entry into that knowledge space for biodiversity. What we're doing within BGE, of course, comes from a very specific point of view, which is how we utilize as much as possible genetic resources to be able to describe, understand, and monitor in the future biodiversity. But this doesn't mean that that's the only data type or knowledge type that is required to be able to do so. So it's extremely important that we establish those infrastructures or those points of communities that own basically or co-own spaces. And through that, we can actually interconnect with other resources. And again, treatments are essential to that. And I hope that, of course, we can do this also through the treatment bank that, yeah.
Thank you, Dimitris. There are two last questions, and I will take them. The first one is from Mark Blackster. The current distribution of sequencing center is concentrated in a few countries. Are there plans to expand investment in centers in additional countries? Personally, I don't know. I will just say that following the global biodiversity framework, many things are being put in place. I know that China, for example, is developing many sequencing centers. But to coordinate all that, UNEP, WCMC, and the European Commission proposed the Global Knowledge Support Service for Biodiversity, really to map what is existing, what is needed, not only on sequencing, but on anything. So this was just launched in December. I'm going to Nairobi next week and the following week for Substar 25, where all this will be discussed. So it's in development. And yeah, very probably there will be more countries joining and hosting sequencing center. And then the last question by Rob Waterhouse. The Knowledge Center for Biodiversity can sometimes feel a somewhat intangible body to researchers on the ground. What are the plans to better connect projects and people to the Knowledge Center? Very good question. And as I tried to show with the slide, it's a complex landscape, but it was even more complex before. We are really trying to rationalize what is important for any researcher in any project is to know that you could be contacted by the Science Service for Biodiversity within the Knowledge Center for Biodiversity. And also, we are discussing a way with BioAgora, coordinating the Science Service for Biodiversity, so that any researcher from any project could also alert the KCBD about big priorities, knowledge gaps identified. Just also to say that at the level of DG Research and Innovation, with my team, we are preparing a master table of any project linked directly or indirectly to biodiversity since 2014. We have already more than 1,000 projects, so we are really trying to analyze that and see the best use we can do from all these projects, and also what we can do with the ongoing project. Okay, I think we have to close. I just want to thank everybody, and also the technical support team, because yeah, you were there. Thank you very much. We were a bit stressed this morning. Well, at least I am. Just really to underline that, yeah, this is a new chapter. And when we speak about protecting and restoring biodiversity on this planet, yes, we need the information provided by genomics. Any specimen we lose is, it's an impact on the richness on this planet. It's normal that some specimen disappear. This is evolution, but also it's very important that we keep many genes for the future. So this was a little bit the message from Richard Dawkins in The Selfish Gene. But we also need genomics when we have to take actions very quickly. It's not only about one species disappearing. Many people will say whatever, but actually it's any species disappearing is one brick in the wall of life, and then some ecosystem could fall apart when some key bricks disappear. But it's also at the level of populations. And this is very, very important. It's not only at the level of species, it's at the level of population, local population. So we need you. We are there to work with you with pleasure. And I don't know if Dimitris, Peter, and Camila, you want to have a last word. But from my side, thank you very much. Very interesting. And let's continue. Let's do it. Thank you. Dimitris.
You are leading this work within the DG RTD. So thank you very much for everything you're doing, and your presence here shows also your commitment to the community. So thanks again.
My pleasure. Peter?
Just reiterating those thanks, and but also the importance of that message of urgency that you conveyed.
Absolutely. And last but not least, Camila.
The same for me. I think it's an honor for all of us to participate together in this endeavor and joining forces, scientific forces, but also from the society and the European Commission. This is going to be the way for us to use science to help protect biodiversity. Thank you.
Well, it's an honor for me too. So yes, we are working as a team, and we have to do it, and we will do it together. I wish everybody a fantastic Biodiversity Genomics Europe Day, and also Biodiversity Genomics Conference this week. Thank you very much.
Thank you.